Welcome everyone to this new ESDR live webinar kitchen, so that I have the pleasure to chair with Sandrine Dubrac from Austria. So I'm Katia Boniface from France. And as a reminder, so ESDR kitchen webinars occur once a month. So we have 4 topics. So the recipe book about new research techniques, the sweet and sour. We are 2 protagonists debate on a hot topic, the freshly baked, who is with presenters of their last high impact research. And so today we have a molecular cuisine, and we have the pleasure to have Martine Bagot that will discuss her journey. So as a reminder, so you can ask all along the talk your question on the Q&A. So please don't hesitate to ask your question. And with Sandrine, it will be a pleasure to ask your questions to Martine. It's not possible to ask the questions live. And so today's webinar is supported by Bristol Myers Squibb. So we thank them for this support. So it is now my pleasure to introduce Martine Bagot from France. So Martine, so you are dermatologist and so a professor of dermatology and the former head of the dermatology department in Hôpital Saint-Louis in Paris. So you are very well known for your work on cutaneous T-cell lymphoma, so both the clinical and translational work in the lab. So you were also the head of the research team on dermatology, so focusing on in cancer and autoimmune inflammatory diseases. So you have been and are still actively involved in societies. So you were a former member of the ESDR board and also in a lot of board of lymphomas, international societies. So you have almost 800 publications, so which is quite impressive. So we are really happy, and we want again to thank you for accepting our invitation to discuss about your journey. So the floor is yours, and you can share your slides.
Thank you. Thank you. So good morning, everybody. So today we are to speak about how identify novel targets for the treatment of cutaneous T-cell lymphoma. Here as my, sorry. And now I have problems to go ahead. Okay, so my conflicts of interest, and today we will speak quickly, very in the brief talk, of a new targeted therapies for CTCL. I will speak about a new, recently approved mogamulizumab, that is an anti-CCR4 monoclonal antibodies, already approved, trial in progress concerning lacutamab, an anti-KIR3DL2 antibody, and 4 possible new targets that will be the subject of the trials in the next few years. Quickly. So mogamulizumab is an antibody directed against CCR4. CCR4 is a molecule expressed by lymphocytes with the skin homing, and it has been evaluated in a controlled phase 2 trial against vorinostat. Vorinostat is an oral inhibitor of histone deacetylase. And as you can see in this paper, it has been shown that mogamulizumab induces a better progression-free survival than has been approved by FDA and EMA after failure of 1 systemic treatment. And we can see, interestingly, with the patient treated with mogamulizumab, a decrease of the tumor T-cell clone, both in skin and blood. And here you can see with high-throughput sequencing that the patient had a dominant T-cell clone, and then after treatment, he has only recovered an oligoclonal distribution of the lymphocyte both in skin and blood. And interestingly, 1/3 of the patients are experiencing mogamulizumab-associated rash. And this rash is characterized by the replacement of CD4 tumor T lymphocytes by CD8 reactive T lymphocytes. And also, as you can see, an increase in macrophage and monocytes. And we did a work on the pathophysiology of this mogamulizumab-associated rash. And you can see that the T-cell clone decreases quickly in the blood. You can see in the middle that the T-cell clone in the skin present before the treatment decreases progressively. And interestingly, on the right part of the screen, you can see that the patient who experienced mogamulizumab-associated rash present a better and increased survival. How is that possible? We showed by high throughput sequencing that the patient experiencing MAR present a recruitment of new non-malignant T cell clones in the skin. Here you can see a patient with pre-treatment had 60% of tumor T cells. You see that when the patient experienced MAR, you see that the T cell clones has completely greatly decreased. And you can see in this as a patient that tumor T cells representing 17%, and at moment of rash, there is a completely disappearing of the tumor T cell clone. So mogamulizumab is a very interesting new treatment. However, sometimes the patient may escape the treatment of the antibody. Why? Because you can see, for example, this patient has a minor population of tumor T cells not expressing CCR4. And you can see that with the time, this population is increasing, and sometimes there is a down modulation of the expression of CCR4. And you see that this patient experienced a complete remission during 3 years, but after 3 years, you see that the tumor T cells progressively reappears. So it is very important to try to develop new other targets. Lacutamab is an antibody directed against KIR3DL2, and we worked during a long time, we're using this antibody, and it is a first-in-class humanized anti-KIR3DL2 cytotoxic-inducing antibody. And this antibody is under development for the treatment of both cutaneous T-cell lymphoma and peripheral T-cell lymphoma. There has been a phase 1 data published in Lancet Oncology 4 years ago, and this phase 1 showed it was performed mainly in Sézary syndrome patients who have been treated by at least 2 prior systemic therapies. And you can see that these phase 1 studies gave very interesting results. And as a result, this drug obtained orphan drug designation for the treatment of CTCL both in Europe and by the FDA, and had obtained a PRIME on Europe and Fast Track designation for Sézary syndrome patients who have received at least 2 prior systemic therapies. Here are the results of the phase 1 studies, showing, as you can see, very interesting results, although not all patients had to complete the maximal dose. And this has led to the design of a phase 2 trial called TELOMAC with several arms. You see a major arm with a Sézary syndrome patient having failed 2 previous therapies, including mogamulizumab, that is the reference center for Sézary, the only approved treatment for Sézary. And there are also 3 cohorts of mycosis fungoides expressing or not KIR3DL2. The drug is administered every week for 5 weeks, then every 2 weeks for 10 administration, and then every 4 weeks. And there are many study endpoints, mainly overall response rate in different compartments, and also PK analysis. And you can see that the interim efficacy result in Sézary syndrome patients shows that there is quite a very interesting responses in the blood and in the skin. And if we look at the response in the blood, you see that there are many patients experiencing CR or PR, although these patients are patients with advanced disease and a lot of previous lines of treatment. And you can see that the patients who present the better responses are the patients who present a complete response or PR. All patients have good responses, but the highest, the expression of KIR3DL2, the highest response, you see that the patient have the best blood response are the patients who have the highest percentage of KIR3DL2 on their blood cells. So this is a very interesting new antibodies, because there is with the mogamulizumab, because of the depletion of Treg cells, you have autoimmunity, you have this rash, you have vitiligo, you have alopecia areata. But with this new antibody, you completely select the tumor T cells. You do not act on the Tregs, and it's really a very targeted monoclonal antibodies on tumor T cells. And you can see also these 2 cases from Stanford. You see that these patients were advanced Sézary syndrome with 10 previous lines of therapy, 6 previous lines of therapy. And you see that all these patients had a very important involvement in the nodes. You see they had NN3. And you see that all these patients experienced complete remission in all compartments, skin, blood, and lymph nodes. And so we are really wondering to have the major results of the Sézary syndrome and mycosis fungoides arms, but it is a very interesting new treatments for the treatment of Sézary syndrome patients. Another clue is the pathway using CD39 and CD73. You see that CD39 hydrolyzes ATP and ADP to adenosine monophosphate, and then they are transformed into adenosine by the ectonucleotide CD73. And adenosine binds to the A2A receptor and inhibits the T and NK responses. It has been shown that Sézary syndrome, in addition to express KIR3DL2 and CCR4, also express CD39. And you see that healthy donors do not express CD39, but Sézary syndrome patient really express CD39. You see that on the right part that it is that tumor T cells that express CD39. And also there is a strong expression of CD39 in the skin. And so it has been shown that in an ATP-enriched cellular environment, the inhibition is more detrimental for tumor T cells than for reactive T cells. And then it could be possible to use ADCC-driving antibodies, for example, KIR3DL2 or anti-CCR3, and to add anti-CD39 or CD73 inhibitor pathways. And this could be a combined treatment by both destroying the tumor T cells and increasing the specific tumor T cell responses. And you can see that the overexpression, you can destroy the tumor T cells by using these antibodies. And in addition, the blocking of the pathway is able to activate T lymphocytes and also the lysis of tumor T cells. Another new target, possible target, is CD70. CD70 is a receptor of the TNF superfamily, and it enables the tumor to escape immune surveillance. And you can see that CD70 is specifically expressed by hematopoietic stem cells. So it is a really specific expression by these cells, no expression on other important cells of other organs. And there has been an antibody, a monoclonal antibodies named cusatuzumab, and this antibody had been engineered to induce an increased ADCC. And this antibody has shown that it is able to restore immune surveillance. And this antibody has been studied in treatment for the treatment of cutaneous T-cell lymphoma. And it has been used in the 27 refractory cutaneous T-cell lymphoma and induced 60% PR with acceptable tolerability. The scheme of administration was the same as used for tumor T cells, every 3 weeks, and it was not optimal for the treatment of cutaneous T-cell lymphoma. The administration is better to increase the cover of the targets every week for the first month and then every 2 weeks. Nevertheless, these results were quite interesting, and now there are CAR T cells that are developed that are being directed against CD70. CCR8 is another very interesting new therapeutic target for T-cell lymphomas. You know, it has been shown that TTCL tumor T cells express several Treg markers and immune checkpoints, such as PD-1. Has been shown that tumor T Sézary cells express PD-1. I've shown you that they express CD39 and also TIGIT. And interestingly, CCR8 is expressed by skin-resident memory T cells and tumor-infiltrating T cells. And you know that it's interesting because T regulatory memory cells are suspected to be the tumor of origin in mycosis fungoides. And so it could be interesting because we could, using an antibody directed against CCR8, deplete CCR8 expressing tumor T cells and only tumor-infiltrating Tregs, and we could not destroy blood Tregs because the blood Tregs do not express CCR8. So we could both destroy the tumor T cells and increase the anti-tumor immune responses. So that could be a double effect, quite interesting. So we show that indeed CCR8 is expressed by Sézary cells and several cell lines. You see that Sézary cells really express CCR8. Also, other types of cutaneous T-cell lymphoma, EBV, gamma delta, and Sézary cell line express CCR8. And we also showed that CCR8 and its ligand CCL18 are expressed in Sézary syndrome patients. And interestingly, the expression of CCR8 and CCL18 in skin is correlated with survival. So this could be also a very interesting new target for cutaneous T-cell lymphoma. And as you can see, also CCL8 and CCL18 are expressed in the skin. And so in the next few months, we will begin a multi-center trial, both in 5 European centers, to evaluate the efficacy, the toxicity, and the efficacy of these new anti-CCR8 antibodies in patients with cutaneous T-cell lymphoma. And finally, the last target is a CD38. CD38, maybe you have mainly you heard about for B-cell lymphoma. Indeed, CD38 is expressed mainly by plasmocytes and not so well known to be expressed by tumor T cells. Nevertheless, we showed that CD38 is expressed on Sézary cell lines. You can see this is a 2 Sézary cell line. And CD38 is also expressed by Sézary cells from patients. And to see that Sézary cells are unequivocally expressing intermediate level of CD38. And interestingly, we showed that some patients have been treated with the mogamulizumab. As I showed you, the mogamulizumab, there can be an escape to treatment by the down modulation of the target. And you see, for example, that this patient lost the expression of PD-1 and CCR4 after mogamulizumab treatment, but there was a persistent CD38 expression. And interestingly, CD38 is mainly expressed by advanced Sézary, that is often Sézary syndrome with transformation, that is a higher size of the tumor T cells. And so this could be a new interesting therapeutic target for patients with advanced cutaneous T-cell lymphoma. And indeed, we showed that if we use an anti-CD38 antibody, is that you know that the most common anti-CD38 antibody is daratumumab, but there's also isatuximab. And if we are using isatuximab in test of antibody-dependent cell cytotoxicity and antibody cell phagocytosis, you see that this antibody induces clearly ADCC and ADCP on patients' blood Sézary cells. So this another new possible target. We also have antibody, daratumumab and isatuximab are already developed antibodies for myeloma. And it will be interesting in the few months, next month, to study also this drug in a patient with advanced cutaneous T-cell lymphoma. And you can see, interestingly, also that CD38 is clearly expressed both in skin and in lymph nodes of cutaneous T-cell lymphoma. And so what is the future treatment for cutaneous T-cell lymphoma? I showed you that recently it has been possible to identify numerous novel targets for the treatment of cutaneous T-cell lymphoma. It has been shown with the mogamulizumab and lacutamab that it is possible to increase specific immune responses, that is to completely modulate the immune system of the patient. And also there are antibodies that are targeted antibodies that induce ADCC and ADCP, able to destroy tumor T cells. It has been for the first time demonstrated the possibility to induce long-term complete responses and control of the tumor T cell clone. It has been shown with mogamulizumab and also with lacutamab. The treatment can be stopped and the patient remain in complete remission. However, they may often relapse after a few years. So that's why it's very important to develop a panel of new antibodies. Of course, it is important to identify the biomarkers to select the best responders. I showed you that lacutamab was efficient both in patients expressing or not expressing KIR3DL2. However, it seems that the best responses are achieved in patients that express high levels of KIR3DL2. And in the future, it will be important to determine the best combined and sequential treatment protocol for CTCL. For example, it would be interesting to have combined treatments, both a treatment that destroys the tumor T cells and the treatment that is able to increase the immune responses, the specific immune responses. And I showed you that there are several candidates among these antibodies. Some of them are doing both, some of them are doing one part, and they could be combined, or they also could be used sequentially. And so I hope to have shown you that we are entering a new area in the treatment of cutaneous T-cell lymphoma, and the algorithm, the treatment algorithm of this patient has been completely modified in the next year, and of course a major new hope for the treatment of these patients. And finally, I would like to thank all persons who participate to all these research, and the patient and their family. Thank you for your attention.
Thank you very much, Martine, for this outstanding and very nice overview of current treatments which are tried to cure the patients with cutaneous T lymphoma. So if you have questions, please add them to the chat part, and we will start asking the questions to Professor Bagot. Maybe I can start with a question while people are starting writing their question in the chat part. I mean, very naive questions actually, because it's really not my field of expertise. I know that CD70 is also expressed on dendritic cells. Do you think that the treatment targeting CD70 can actually impair
Immune
TC function, yeah, and then actually be counterproductive at some point of the treatment?
It's a possibility, but I think it has not been shown in the previous study. There had been a study with monoclonal antibodies and preliminary study with CAR-T, and I am not aware of this type of side effect. Yeah.
Okay. Another very naive question as well about this CD70. I mean, as far as I know, it's more related to Th1, and cutaneous T lymphoma, you have more Th2 environments. So how do you explain the efficacy of CD70 in a Th2 context?
When you know CTCL are very plastic, there is great plasticity. Actually, there are many Th2, but also Th1. It's not when you look at the phenotype of the cells, they are very varied phenotype, and it's difficult to say that it's impossible to say that the cells are only Th2, and also you can have aberrant expression on cell Th1 with also aberrant expression. So I would not focus on a specific subtype of cells expressing. We see a various range of expression of atypical molecules in cutaneous T-cell lymphoma. Yeah.
Okay.
Maybe I would ask,
yeah.
Sandrine, I had few questions also myself about because it's interesting to target CCR8, because at least that's what we see in human skin is that usually the healthy T cells that express CCR8 do not express PD-1, so that's exclusive. And one thing I was wondering is whether the treatment of patients with mogalizumab would lead to a downregulation of CCR8 expression, either on malignant cells, but also on healthy cells.
It has never been studied. We do not know that. Actually, until recently, there were very few anti-CCR8 antibody working on immunohistochemistry. Now there are some antibodies, but it's very recent. I am not aware of such. It hasn't been studied in patients treated with MOGA. Yeah.
Okay, because because you were mentioning with mogalizumab that at some point, so you have resistance to therapy that is happening, and I was wondering whether it could be a good opportunity afterwards to treat patients with an anti-CCR8 or not, or whether you could have it could be linked to that, whether it would be because CCR8 or other molecules that you are saying like CD39 or CD38 are not dampened also with the treatment.
We already know that patients having down-modulated CCR4 can be a respondent to lacutamab, anti-KIR3DL2, and I would guess that that anti-CCR4 does not down-modulate CCR8, but that monoclonal antibody directed against CCR8 and another therapeutic options in patients having escaped anti-CCR4, eventually anti-KIR3DL2, and in the future we'll have a panel of new antibodies, and we could use them sequentially when the patient becomes resistant to a first or second line of antibody.
Okay. And after, did you look for CCR8, because at least for Sézary and mycosis fungoid, what they showed is that the malignant clone, for one disease, would be more like a recirculating clone, whether it's more a resident clone in the skin. And I was wondering whether there was a differential expression of CCR8 depending on whether you had a recirculating clone, whether a resident T cell clone in the disease.
No, actually, CCR8 is expressed by most cases, more advanced cases that would be recirculated, maybe recirculated, but it is not so much linked. It is mainly linked to the expression is higher in more advanced cases. However, you know that again, Hélène Moise showed that it was said previously that TCR that Caesary cells were central memory and the other resident effector memory, but it's not true because when we studied the Hélène Moise and the other teams looked at the expression, the phenotype of Caesary cells, and actually they are much more heterogeneous than previously thought.
Okay.
And there are central memory, but there are also resident memory, there are also naive T cells, so it's not so simple that we thought at the beginning. Yeah.
Okay. Maybe I will ask a last question is so because it's a good opportunity potentially with all these treatment like and trials that are coming up. So do you foresee from these molecules and one that would have maybe like less side effects or that would work better than the other ones?
Well, actually, the anti-KIR3DL2 has the less side effects because when you look at anti-CCR8, we know that there has been already trials in patients with a solid cancer, and actually they had rash also. I thought that patient would not have rash because it doesn't touch the Tregs in the blood, but actually they are really doing rash like CCR4. So the antibody with the less side effects actually is anti-KIR3DL2.
Okay.
Okay, thank you very much, Professor Bagot, for this very interesting discussion. I think now we are getting close to the end of this webinar. I would like to remind you the next webinar that will take place on the 16th of October. It will be a sweet and sour webinar on microbiome, and it will be the discussion in between two experts of the field, Hélène van den Bogart and John Common. So you are all welcome to attend this webinar as well. I would like to remind you that all ESDR Kitchen webinars are available on YouTube. So for this, you just go to the ESDR website, you click on the ESDR Kitchen link, and then you will have all available webinars, the link to YouTube, and it's for free. So please do not hesitate to watch them as much as you want. So yeah, the next webinar will be on the 16th of October, and we all thank you for your attendance. Katia and me are really pleased to have done this webinar, and we really warmly thank Professor Bagot to have attended and to have given this excellent lecture. Thank you very much.
Thank you very much. Goodbye.
Goodbye.
Bye. Bye-bye.